YouTube, and I think one I forgot. Oh, doesn't matter. Doesn't matter. So let me just check if we are live and if everything is going well with the streaming services. Yes, looks good. So
okay.
we're already 13 seconds into the streaming, Thomas. Okay, got it. We also got a nice message here. This meeting is being live streamed, that we just start talking right away. Thomas, it's very good to see you again after the bio.
Yes, absolutely, Christian. It's my pleasure to be here. Really great opportunity, and yeah, nice to meet you as well. So as a person, it's really nice.
It's already over a month ago, isn't it?
Yeah.
But time flies.
Time flies. It's amazing. It's incredible. But it was a good conference, I think.
How was it for you? It was the first after 2019 in my life. How was it for you?
Indeed, at least it was not the first after COVID for me. So I was already at other conferences, but not that size. And I had a very good feeling that a lot of people were very kind of relieved to see people again and to interact and to talk and just to chat, even just random without any scientific or business related topics, just have a nice chat. It was really amazing. I really enjoyed it.
I couldn't agree more. It was my first bio after 2019. I think the other bios in Europe, Bio Europe, so we're pretty much virtual. And this year, the first one was in San Diego. In June, which was a little bit strange because the airlines were not at the standards that I was used before the pandemic, which was quite funny. But I absolutely agree to what you said. It seems to me that everybody that I met is pretty happy that we have real life meetings back again. It is like meeting old friends after 3 years. And interestingly, I mean, before the pandemic, I expected that this digital turn that we got in March 2020 will just vanish and will just go away. And I was very, how should I say, pleasantly surprised that it doesn't change. So a lot of pharma companies, a lot of VCs have integrated now digital versions, like also this webinar, for example, into their processes. And I can highly recommend to use these new tools because people used it for scouting. What is your experience with webinars?
Absolutely. I fully agree with you, Christian. So we are quite active here, also at Tuwind for webinars, indeed. So we've done quite a number of webinars with partners to show what we work on and to also to educate people, because what we realized a lot is that the way of how people look for things, how they search for things in COVID has completely changed. Before, and I think it's now back again, but before, you were meeting at a conference and had a handshake, a good laugh together, maybe some beer or wine, and things came along, right? And then suddenly this hard stop was there, right? You couldn't meet anybody, and then everything was towards Google, Yahoo, Bing, all those different things, and so this was of course also a chance for such a company like us to also use social media more and more. And if you say that webinars is also social media, I would also say that we are very active here. Yes.
In my life, everything is social media. I mean, right? So everything online is just social media. I agree, because I mean, before the pandemic, I always tried to integrate live streamings, webinars, video calls into my business life. I love games, so I know these tools since the 90s, and also did research with Harvard, University of St. Gallen to find out how the internet will transform our businesses. But in the pharma industry, nobody used it. So every time when I said, let's do a webinar, they said, no, why not? Why? And now, the last time in the bio in Leipzig, I asked people, okay, what do you do when you want to reach out to a company in the conference system? And the majority answered, when I find a company that I think is interesting, I go to Google search. And I want to know more. So
that's true, absolutely. So we can see that on our homepage as well, on the click rates and these kind of things. They dramatically increased in COVID times. And it's good because our homepage is also quite having a lot of content. It's really educational. People can not just learn about us, but really about the topic biophysics here. And so people really use it to educate themselves, which we really enjoy because that's also part of what we do. Right?
So let's use this webinar to educate me, and let me ask the first stupid question. Whenever I think about drug development, what comes to my mind is chemistry, is biology, and every time when I think about, for example, Elon Musk with Tesla, I think about SpaceX, space travel in terms of physics. And when we first met, you explained to me that you use physics for drug development, and there was why physics? Why physics in drug development? Why not chemistry?
No, I think first of all, I think it has to be seen as a global part, or let's say a process where different scientific aspects come into line. And of course, chemistry is highly important, no doubt. This is the basis of a lot of things. But also, indeed, biophysics plays a big role in drug discovery. So this is something that is even now, let's say, further pronounced. So actually, biophysics has gained quite some momentum, I would say. The importance is increasing for several reasons. And I mean, just to define that a little bit more, so biophysics is, I do understand that a lot of people feel comfortable when they read up the first part of this word, bio, it's all like comfortable, it's animals, it's trees, it's light, it's nice, and then physics. Shit. But actually, it's a really cool combination because you use the physical measurement, the physical interpretation to understand the biology. It's really fascinating. So it's really, sometimes when you are going really deep into a detail of a molecular mechanism in a cell, you find it so fascinating. And then sometimes you think, wow, that's just a wonder here. What are we living in? This is amazing. And so you use physical phenomena to understand what the biology does. So that's biophysics. And as you might see, I'm a passionate biophysicist. It's really the thing that I like in my life.
Yeah, let me just share the screen. It usually takes a little bit of time. I just opened a few websites. There's also a biophysical society.
Good society, by the way.
Yeah, what is the society all about?
The Biophysical Society is basically really consortium of well-known biophysicists giving the whole area a certain structure. Obviously, there are experts all around the globe, and this is a network of really the best of the best of biophysics. And you can see here, it's actually a very important society, fantastic, very nice meetings for specific biophysicists applications. And here on the left side, you see already a big topic that has drawn a lot of attention is, of course, as we all know, COVID-19. And so the fascination also from the biophysics perspective is, of course, here also shown to understand how that really terrible virus is entering a cell, how the interaction of this virus with the cell is happening, how fast it is going on, how strong this interaction is. This is biophysics. You read out these factors. That's pure biophysics.
That's good. That's good to know. And this is more for the specialist and for the generalists who want to to know a little bit about everything. There's also Wikipedia with a nice definition of biophysics and physics, the fundamental science on the right side.
Yeah, absolutely. I mean, we use physical methods. Absolutely. We apply them to understand biological processes. Yeah.
Thomas, let me ask you the second question. How did you find this passion? What is your background?
Originally, I'm a biochemist. I was trained as a biochemist some 15 years ago, I guess. And during that time, I got in touch with biophysics at a very specific scientific topic. And I got simply fascinated by the fact that you can directly, by looking at a small detail, learn about a large process. So this is really something that was drawing my attention, and I was fascinated that you can apply physical readouts to really understand a cell and an organism. And so, or at least parts of an organism or parts of a cell. Of course, it's a extremely large, complex system, a cell, but you see and you understand a certain aspect of it. And that was absolutely my fascination. And I got stuck to it at some point because I really like it.
So is physics a huge part in biochemistry, in the education at the university?
Not really, I have to say. So there is, of course, physics, because physics are important, but biochemistry, education, and also, I mean, originally I was a biologist and made a PhD in biochemistry. I have to make it more correct, right? Because this is the real situation. And as a biologist, there were certain lessons in physics, very low level. And of course, in the education, in a PhD, there's not a real lecture, let's call it. So you have to make your own efforts to learn about different other life science aspects, such as physics. And I got quite interested in these things, and it was a lot of course, asking colleagues and other experts and get information from those guys, and they taught me basically.
So physics, biophysics, basically, or physics is a small part of your education. How comes that you then decided right after the university not to work in the industry or stay in academia, but found a company that revolves around the topic biophysics?
I think, first of all, indeed, there was a passion for biophysics, even though it was not a large part of my education, indeed. But it really fascinated me. And then, in addition, there's a second fascination with it, which is actually kind of the entrepreneurship. So this idea to build up our own topic and to make decisions which have a consequence, positive or negative. Everything is kind of what you decided and you have to think about. And that followed my complete life. And I always, as soon as I had situations where I could build up something on my own, I did it, at least in a small scale. And I thought, okay, I could go to other companies. I had also, already, I was quite far with some contracts, but then this chance came along to really build up a new company from the scratch as a startup, as a first employee. And this was something, honestly, if I wouldn't have done that, then I would have always thought about, why didn't you do that in the past? Why did you leave that chance? So I had to do it. And I'm absolutely happy about how things go on.
How did you find the opportunity?
Honestly, I sometimes in life it's a combination of being at the right spot at the right time. I had my PhD supervisor is a very open-minded person. He's very into technologies, and during my PhD, there were certain aspects about biophysics which we could not solve with conventional methods. And he found some person in the physics department at the LMU in Munich, in like the Ludwig Maximilian University. And those guys were designing a brand new technology. And a little bit for desperation, we just thought, okay, let's give it a try. And suddenly 2 weeks later, we had indeed, values and data which even made sense. It's not just random data, it really made sense. And this was quite fascinating. And we all were like thinking, wow, that is impressive. We wasted a lot of time before, and then within 2 weeks, we had cool data. That was cool. And here the chance was growing. So basically, Tubind was a spin-off from the university here in Regensburg in combination with a company called Nanotemper Technologies. That's a company in Munich. It's a producer and developer of biophysical methods. And at that time, 2011, so more than 10 years now, I had the chance to be the first employee and getting promoted to be CEO quite short after that. And I'm very happy that I did this, I have to say. And it was a good chance. It was, of course, a wild drive, let's say. but it was really, really cool, I have to say. Yeah.
Yeah, before we get into the wild drive, I'm a little bit curious about the business model. So
Yeah, sure.
Biophysics, it's not very, let's say, it's not a huge part at the university, I would say.
Yeah.
And you started in a company as a first employee, got promoted to a CEO quite after. And I'm just curious, how challenging was it to find a business model? Is it your focus now with your company with biophysics in drug development and drug discovery?
Yeah.
How challenging was that to establish that on the market? Was it a no-brainer to say everybody knew, yeah, you need biophysics, so let's just work together with you? Or was there some groundwork that needed to be done?
Okay, yeah. So first of all, it was a challenge. I mean, biophysics at that time, so 10, 11 years, was even much more, let's say, or let's say less important than today for several reasons. And in addition, there was, of course, already some competition in that market. And the original idea of 2bind was actually to be a service division for nanotemper technologies. That was basically at the very beginning the idea. Really to have an entity that is using these devices to also educate people and show what these devices can do. Indeed, actually, this has changed. So in 2017, we sat together and decided that 2bind is getting independent of nanotemper. So we're a completely independent entity now. And the main reason was that we wanted to develop the company in a different way. We are in the moment, or we wanted to become an expert niche player in biophysics. We are working on a fee-for-service business model in the moment, also starting the first partnerships with interesting companies, or actually these guys realized over the last 10 years that we have quite some knowledge and quite some capabilities, and now people approach us also with partnership requests to develop something together. But our fundamental principle was to be a service provider. And this has actually a very simple reason. We never had an investor. So where to get money?
From customers.
You need to get money in, right? You want to develop the company, and without having any investor in here, you needed to have a business model where there's a fast cash flow and where you can get money in and develop a company. And that was actually also a big part of the decision to use that kind of service business model.
Yeah, the traditional business models that I learned at university and in a commercial school before was find customers first. And when you want to grow your business, then you talk to investors. This new world like we have now, where investors invest in startups without any business model, was quite unusual in Europe, at least. In the 90s or in the 80s of last century. So it's a smart move, basically, to find customers. Let's get a little bit into investigative podcasting. You mentioned there were for certain reasons biophysics was not so important 10 years ago, and its importance increased in drug development and drug discovery over the last 10 years. Why was it not important, and why did the importance increase? Two questions.
Yeah. So first of all, one has to say that the way of developing drugs and discovery of drugs has been changed since the last years. So 10 years before, a lot of the targets that were out there and were worked on were enzymatic targets, which had a clear activity that you can read out with biochemical approaches. A biochemical approach is different to a biophysical approach, since you use an activity that the protein has to generate a signal that you can read out. Biophysics is more interested in the direct interaction rather than the activity. And so a lot of the efforts were done, first of all, on biochemical approaches. So HTS campaigns, meaning high throughput screening, was done on a complete different, let's say, platform, right? It is a different life science part. And biophysics was only this tiny little piece when you had your compound, and then you saw an activity, and then it's just like this confirmation thing, like, will you please confirm that this one thing is doing something? So it was a minor part, let's call. Of course, I'm a little bit over exaggerating, but my.
But it's always good for podcasts.
Yeah, I mean, you have to make it understandable.
And
of
fun.
Course,
science can be fun as well. So.
Yeah, of course, of course. And yeah, sure. I mean, biophysics also played a role in the past, but nowadays, it is, for example, the case that hard to drug targets in a cell, they cannot be read out with a biochemical approach. There's no other way than a direct binding event. So you need, when you want to develop and discover a drug, you need to find a binder which binds to the protein or the RNA, whatever target you're interested in. And since there's no biochemical approach, you need biophysics. You need to see the interaction. You need to understand how fast this goes on and off and what the affinity is of the two. And therefore, the attention that is now for biophysics has increased. And in addition, not just the target side, so it's not just new protein types and even a complete new field, the RNA as a target. So it's really going away from the protein towards an RNA as a direct target for a small molecule. For example, you want to establish a certain structure in an RNA or disrupt the structure. And as a consequence of that, the cell has, is somehow modified, whatever aspect of a cell you want to modify. So this was not there 10 years before. This is a relatively new approach, and the complete approach is solely is the wrong word, but heavily depending on biophysical approaches. And so a complete new field emerged even that was not present at 10 years. And then in addition, if you always have two molecules for a drug development, the target and the drug. And before it was always like small molecules, some 300, 400, 500 Dalton in size, had certain rules to fulfill, and basically you were constantly screening things and making efforts if the cell is somehow affected. Nowadays, it's not just this one single small molecule that is in focus. Now, complete new entities come along. For example, PROTACs. Yeah. So this new class of molecules has a complete different approach than a classical inhibitor. So it's not an inhibitor. So a PROTAC is a combination of two smaller molecules which are linked with a linker. And the idea is to bring a protein of interest in the close proximity of an enzyme that tells the cell to degrade that protein. So the cell is told to get rid of this one specific protein. This complete concept was not present 10 years ago. And again, we're not talking about an activity. You need another readout, and binding is one of the interactions, or one interaction is one of those things that you can read out with biophysics. So a long story short, many different changes in the environment of drug discovery have come along in the last 10 years, and all of them kind of push biophysics. And that is the reason why now biophysics as the application, but also the outsourcing market is increasing constantly. And of course, for us, that is something very nice. And we were happy to realize that new entities come along, new targets, new things that we can address. And that allowed us to build also our portfolio to serve the customer in the best way.
That's great. That's great to hear that drug discovery is changing, and it's getting. I think it's also getting more and more complex, isn't it?
Oh yes, absolutely. So this classical paradigm: you have one protein, you tackle that protein, everything is solved. No. So nowadays it's really a systematic approach. You need to consider a lot of more things. Honestly, the very low-hanging fruits are already gone, and now it's really there are absolutely fascinating concepts out there. Again, we're in the lucky position because, sometimes, let's say, when challenging things happen, it is sometimes good to start with an outsourcing approach, giving an expert a first try and then integrating things into own labs. And therefore, we get in touch with a lot of state-of-the-art or really at the edge ideas. And that is what is really fun here. So we're confronted with projects which are super innovative and super challenging, but that's the fun. That's really cool.
Is your position, what are you doing with antibodies? Is this also a part of your business?
Yeah. So let's make it a little bit different. So biophysics can serve to answer two types of questions. One type of question is related to the interaction of two molecules, A and B. So they bind fast and slow, or they bind strong, or there are two of these and just one of those, and so on. So it's binding parameters, first question. And the second question is, for example, for antibodies, it's very relevant to stabilize an antibody. If you imagine you have a drug and it's in a syringe and you want to push it into an arm of a patient to cure the patient, the antibody has to be very stable in there, because the vessel or the syringe or whatever, the solution has to be stored somewhere, 4 degrees or whatever, in a liquid formulation. And this is something that you can really nicely address with biophysics. So we serve not just those guys from the drug discovery area with the interaction analysis type of things, but we also work a lot on antibodies and determine the optimal storage conditions of an antibody. Let's make it in simple words. A very important point as well, but very different, because the one thing is very early. R&D, super, sometimes very, okay, wow, that is a cool concept, that's fascinating, let's think about it first. And finding, the formulation of a well-established antibody, which is already super characterized, it's a complete different aspect of what we do, yeah.
So we can also add value to established technology that is already on the market, make it better, improve it, understand it better. Then, so basically, you look in detail on how things work.
Yeah, exactly. As mentioned before, we use the physics phenomena to really check out what's the exact mechanism of something. And I think there are so many applications, and we're so happy to be at that point, because we made absolutely fascinating measurements for example, for in vitro diagnostic guys, where we helped to develop tests for an allergen in milk, or an allergen in apple juice or something. So this is really fascinating. This can also be solved with biophysics. So it's a very universal approach, actually, biophysics.
One term came across my table in the last 2 to 4 years. It's ADCs, antibody drug conjugates. Does biophysics also play a role in that?
Yeah, for sure.
Which role? So next question, which role?
Yeah, and so
I'm not satisfied with for sure.
For those guys who might not know ADC, so it's basically the idea is that an antibody, which is a molecule that can bind to a certain target structure. So it recognizes a certain structure, for example, on a cell, for example, on a cancer cell, let's call it. There's a cancer cell, and the surface on the cancer cell has certain structure, certain function, and an antibody is a binding molecule which recognizes the cancer cell, for example, but discriminates native cell. And of course, the idea is, if I add a payload to the antibody which is toxic, then I can toxidate the unwanted cell in a cancer and leave the native environment untouched. So this is an ADC. And of course, this payload or warhead, a lot of people call this warhead, has to be attached to the protein, so to the antibody, via chemical linking. And of course, that chemical linking is a modification of the original protein, right? And again, you need to understand, does that influence the antibody or not? Is it still doing what it should do? Is it still binding to the target? Is it still, stable? Does it fall apart? Is it suddenly, does it tend to aggregate? Does it go out of the aqueous solution and is then, of course, not a drug anymore? These things we can also work on, yeah.
So we can really measure it and get more details on that. I mean, I find the concept very interesting. It always reminds me a little bit on about warfare. It's like an aircraft. So you put a payload, missiles on an aircraft, and then you send the aircraft into the air, which basically in ADC terms is into the body. And they always had a hard time to understand, I mean, how do you guarantee that the payload is released at the point of action where it's intended and not somewhere else in the body? How can you measure that? When it's in the biological system? I think it's easy in the lab, so to say, okay, this happens and that happens, but a body is different. How do you measure that in a body?
First of all, I'm not really familiar with the body. That's maybe also something. In general, the biophysics guys, we don't really work with a living cell, and we don't have that. We are completely in vitro guys. But I know that there are phenotypic readouts where you can realize if a cell is reacting on that antibody, if it changes its shape, if it suffers. You can see that when a cell changes its shape or even dies. So you can see that. Honestly, how they measure this in a living body, I have to admit, I have no clue.
So your work is basically in vitro. So it's basically on the lab bench before clinical development.
Exactly. Correct. Absolutely.
Where is your company positioned in the value chain? Let's talk about your organization.
I mean, to mind, of course, first of all, we are outsourcing partner, and outsourcing can be done at different position of the value chain. So it can be in the very early phases in classical drug discovery, for example, when you need a hit identification, meaning you screen large libraries of compounds. We do have own libraries. We can also work with external libraries and identify potential drug candidates. Then the next phase of the drug discovery phase is the validation of potential hits and drug candidates. So this could be that a customer has already done internal hit identification and then wants to get a third-party independent answer. Is that a real hit or not? Is that a real binder or not? And of course, then commonly these small molecules are optimized in the medicinal chemistry approach. So basically, you want to understand how the structure of the complete protein with a small compound drug is, and then optimize that. And here, of course, again, biophysics is relevant because we monitor the evolution of those compounds in terms of the binding to the target protein or RNA. So this is at the drug discovery level. So this is the first very early drug discovery R&D.
So sorry to interrupt here. So basically you start with really basic research. So you're a partner for the basic research organizations initially.
Yes, correct.
So it's a lot of universities, basically, also basic research within pharma companies.
Yeah, so I mean, I can maybe describe with some words our clients.
Sure, go ahead.
First of all, I think very obvious answer would be everybody that needs biophysics. Obviously not what you wanted to hear. We are working largely with biotech companies, VC backup biotech companies from US and also Europe. So these are our main business partners. We work also a lot with pharmaceutical companies. Since 2011, we have now a portfolio of customers, around about 450 customers, and we have worked on around about 1,400 projects with them. So in the last 11 years.
Impressive.
Yeah, I think it's really a good track number. Yeah.
You said there's an uptake in biophysics. Do you also see that in the numbers of projects that you get, that it's increasing year over year?
Yes, definitely. But I'm not sure if this has to do with the increase of biophysics or because people now know us and realize that we have a certain competence. I think it's a mix of all those effects. But yes, constantly increasing. I mean, this, of course, also on the key parameters on the financial basis is also, you can see that, yeah.
So we have drug discovery. What's another part in the value chain where you can assist?
Yeah, so basically the other part where we can assist is, for example, in antibodies, in the antibody development part. It starts with a classical developability study. So the idea is an antibody is generated, it has an activity, it has a binding event, but this does not mean that this will be translated into a drug because certain physical properties are not good. And a drug should have all optimal parameters. And here we offer services where people can check their candidates for those parameters and then select the ones that have most promising parameters and get them into the later phases. So this is preclinical, let's say, in that area a little bit earlier. And then, of course, an antibody, as I mentioned before, has to be at some point administered to a patient. And before doing that, you need to have a proper, buffer condition, a proper storage, which is commonly called a formulation of an antibody. And here in this formulation development, we can also help with quite some nice tools that we have developed over the years. Yeah. So it's a very late stage offer that we also have. So early, somewhere in the middle, and also late.
I was just smiling because you said ultimately the drug or the antibody should be administered in a patient and hopefully help the patient. I felt it in my mind, but I completely agree to that statement in drug development. So we need a patient. I'm not so sure in drug discovery or research organizations. So very often I think it's more about finding something new.
Yeah, absolutely. So that's a complete different concept. Yeah. It's finding something new versus you have something interesting and you want to optimize. Yeah.
Which I find very interesting because, I mean, in my opinion, this is one of the complexities in drug development, that on one hand, within a company, investors and leadership need to work together with creative people like scientists who needs to find something new. But on the other hand, you also have this project management approach where leaders and investors need to focus the team on one single goal, which is a little bit contradicting in my mind. So it's either you are creative or you are goal oriented. How do you see it? Bring this right together.
Yeah, so I think I'm with you on that point. So there's definitely something difficult around that. I would say. I think in general, this is a very big challenge for drug discovery, but also for late stage projects, that the tasks are so different, and also the people that are working on these things have different mindsets. So or should have, let's say. In a very early drug discovery phase, you need to have a certain space of creativity, because it's not everything. There's no SOP that you can follow and then the standard operating protocol will tell you what the outcome is. This is not possible. You need to be open-minded. Of course, for VC backed up companies, there's a certain core set where they can act for a different reason: strategic, budget, resources, and so on. And the creativity should not be limited too much by these external factors, because you need that creativity. Whereas when you go for late stage projects, it's more defined, and still creativity is needed, but a different type of creativity, I would say. So yeah, I see where you're going to.
No, for me, it's very challenging to integrate different personalities into one value chain to move things forward. Because I mean, creativity in later stages, and just think you mentioned SARS-CoV-2, for example, mRNA in the beginning when we were looking at this Institute of Biophysics. 40,000 patient studies is a huge task. So being creative within a 40,000 patient study wouldn't produce any results that are useful to talk with the regulators then afterwards. On the other hand, when we would try to put a course set around the researchers at the universities, who needs to identify new approaches, for example, new targets, new chemical compounds, and just run them through SOPs and say, okay, you need to do this procedure every day. I think it would suppress creativity.
Absolutely. I fully agree. I think for the different positions, there are certain characters needed and certain people mindsets. And as I mentioned, I think everything has a right to be there and has a reason to be a valid partner for these and these things. Absolutely.
Yeah. How do you handle these challenges? Because I mean, on one hand, you're a partner to research organizations in the early stages, drug discovery, also on the drug development side. How do you handle these challenges within your company?
Yeah. So first of all, one has to definitely educate our project leaders, which are the first communication line on the scientific level to the partner that we have. and emphasize what the intention of the respective colleagues from the other end is. And obviously, the intention of an early drug discovery guy is, or a person is different than late stage development. And people have to understand what the really need is. For us, honestly, this is a very important point already at the very early beginning of a discussion. So we really sit down together with a partner, with a client, a customer, or a collaboration partner, and sit down and define what is the aim. So we need to know that. I know that a lot of other CROs are not interested in that too much. But I also know that others also see as we do. I think we need to know that to understand what the need is of a late stage or an early drug discovery guy, to really understand what the focus of the project should be. And therefore, intense discussions are needed to get a sense what's the aim. And then you are on one level, and then you understand, okay, this is the need, here we can serve and help.
Yeah, project management, I think it's very important, and also to get the goals and the targets and the aim of a project out of people into a useful process at the end of the day. Yeah. We need to deliver results. The market is big. We have 8 billion people on the market. Now let's continue with investigative podcasting. Why are you the best experts in biophysics?
So first of all, I think that there are others out there which are good. So I don't want to say something else, because that would simply not be true. There are other companies out there and other groups which are doing an excellent job, no doubt. I think our company, TwoBind, is a little bit different for different reasons. First of all, on the market, you will not find a company that is dedicated solely on that niche of biophysics. This is our core expertise, and we're doing only biophysics. Biophysics and other competitors, let's say, it's a part of something. Most of them are one-stop shops. Again, I think they do a good job, but it's a one-stop shop. It's a completely different concept. We are absolutely the opposite. We're not a one-stop shop. You get biophysics here, and I think quite good biophysics. That's what we want to be. We want to be experts in this niche of biophysics. And so already at the description of how we see ourselves and others, there's a certain differentiation. And then there's, of course, how you can prove that you're an expert. And for example, one thing is, for us, it's very important that we have technology partners. So we back up with technology partners. Nanotemper is one of them, Creoptics, now Malvern is one, Dynamic Biosensors is one. And these guys, we are very close to them. And this means really that we are beta testers. We get devices in here at TwoBind which are maybe never reaching the market, or maybe they do. We are testing this. And it's a very fruitful give and take. We learn a lot about new technologies, about new approaches, hardware, software. But we also teach what the application in real life should look like, because that's very often the thing that developer of a device are simply missing, the sense of why you want to do that. And so it's a very fruitful combination that we have with those guys. And therefore, we indeed have some tools here at TwoBind, some of our 11 technologies in TwoBind, they are customized. You cannot buy them on the market. And in addition, of course, because we get a lot of knowledge from the exchange with those partners, which is, of course, not accessible to others out there. So I think this is a reason why we can, at least to some extent, say that we're experts, I guess.
Now I'm curious. I know we didn't have prepared a presentation, so it's a podcast, it's a conversational format. But you mentioned that in your company, you offer 11 technologies to your customers. So which technologies are these?
Yeah, okay. So first of all, let's start with the technologies which look at proteins and how these how proteins look like. So structure and not structure, but molecular weight, size, all those different aspects, colloidal stability, chemical stability, thermal stability. So this is the classical SEC-MALS. This is dynamic light scattering. This is PANTA. This is nano-DSF. So these are the tools that we use to characterize proteins. Then on the side of basic equilibrium binding methods, we do have spectral shift, a brand new technology of nanotamper. We do have microscale thermophoresis in different variations. We have an automated, which is a HTS capable device, which we have a dianthos, which is even more HTS capable. On the kinetics level, we do have a classical characterization format for biologics, antibody antigen, called biolayer interferometry, BLI. We have a very fascinating technology called wave delta. It's from Creoptics, now Malvern. It's a great coupled interferometry method for kinetic studies. Brilliant technology anyway. And the latest technology that we also have integrated is the dynamic biosensors helix system, which is an awesome tool for PROTAC determination. So ternary complex formation is brilliant. And of course, we also do have gold standard methods like isothermal titration calorimetry in-house. So these are the technologies that we offer. I hope I mentioned all 11. And of course, in addition to these methods, we of course need to have a certain equipment in the lab anyway. So pipetting robots, liquid handling stations, lab side ecosystem, for example, we need to have them. Otherwise, we cannot, create or get all the capacities and capabilities out of the devices that we have.
So you know all your technologies by heart, basically.
Yes, that's what I think an expert should do. Yeah.
Yeah, absolutely. So this means that anybody who wants to work with you is not only tapping into expertise, but also technology that in that form is not available on the market somewhere else.
At least parts.
In this combination.
In this combination, I doubt that you will find another company anyway. But important for us is always that we see ourselves not as a platform company. We do not want to be a platform. So a platform is a very efficient, way to approach things. You have standardized, miniaturized, optimized processes, and it's very fast, efficient, and so on. But it's not flexible. And as I mentioned before, a lot of people come with brand new ideas to us, and we cannot work with a platform. It's like having a hammer, and then somebody's coming along with a short nail or a long nail, and you drill it into the wood. But what happens?
Use always the same hammer.
Exactly. It's a brilliant hammer, very ergonomic, and it's really good in the hand, and you have a good tip, and it's great hammer. But what happens if a screw comes along, right? What do? You need a screwdriver, and the hammer is not enough. And that is what we want to do. We want to have all the tools that we can grab into our box and take out the right tool for the question that a customer has. This is what we want to do. And this is where we are, I think, on a good track.
There was this, I don't know if you translated right, there's this proverb that to someone with a hammer, everything looks like a nail. If I don't know if it makes sense in English. Just popped up in my mind when you mentioned the hammer, the hammer story. So you're not that way. So basically you customize the solution to the needs of your customers.
Absolutely. I think that is a need. Because when you look at the variety of targets already just on the protein level, protein are so different to each other. If you compare a soluble kinase to a membrane protein, they have basically nothing else than the amino acid sequence maybe in common. Anything else is different. And so you cannot use just a hammer. You need a screwdriver or you need something else. And you have to identify that you need the screwdriver in this moment. And for that protein, you need the hammer. That is the idea of what we do. And that is also the reason why we have 11 technologies now in-house. And we're constantly screening the market for new things where we can add more information to what we do. That's the complete idea of Toolbind, being an expert. And consultation is a big part of what we do.
So it's basically very helpful for biotech companies who need the services from time to time, who need an up-to-date toolbox, but don't want to invest right away in building a team, buying the tools, and developing also the tools further.
100% correct. Yes, and I think it's there are a lot of companies out there that are now well-known biotech companies with great success, and most of them have indeed been at some point in contact with us. We have a large portfolio, and we identify tools which they might need also on a daily routine. And then we even support them in integrating these devices at home in their in-house processes. This is part of how we see our function and job. We support them at any level. And specifically for early biophysics, or not biophysics, but early biotech companies, this is, of course, a great possibility indeed.
Let's go back to the entrepreneurship challenges and successes and of your company. What two successes and two challenges? What were your two biggest challenges in the last 10 years that you solved as an entrepreneur?
I think the highest challenge was to get the right people into the company. And I keep it with the sentence that I think Steve Jobs has claimed: you should always employ people that are better than you. And I can proudly say that a lot of people are better than me and know more and more experts. And I'm listening and learning. And I think that is something that was very tough to achieve, to find good people. Biophysics is not something that everybody, as we discussed in the very beginning, does from the beginning on in all education. It's something special, and to find educated people that are good in what they do is tough. And I think that was one of the largest challenges. And of course, other challenges is of course, as an early employee and also shareholder of the company, you always are like 5 steps ahead in your mind. But you know, in a organic growth company, the steps are small, and you would like to, you get kind of, I want to make the next 2 steps now, and I don't want to wait. And so it was also a little challenge to manage the expectation of myself and also about some of the other guys here.
Yeah, walking away, like this martial arts term. So martial artists talk about walking away, or Japanese people in general talking about walking away. And I couldn't agree more to what you said. So the mind is always very quick with the solution, and then there is reality, which moves a little bit slower.
Absolutely, yes. And so many hurdles are jumped suddenly out of the box. Suddenly there's a hurdle you have to spring over. You never expected that this will happen. And of course, that's entrepreneurship, but that's also the fun about it, right? I think being reactive and solve solutions or find solutions for problems, I think it's something that people that love entrepreneur also love. That's part of it.
And they also liked what you said about Steve Jobs, that leaders should always find employees that are better than themselves.
Yeah.
It also reminded me of another proverb. That was, when you are the smartest person in the room, you're in the wrong room.
Yeah, yeah, absolutely. So I think it was Steve Jobs. I'm not 100% sure, but I really love that sentence. But the one that you just mentioned is also very good, because it makes the same consequence, basically. Surround yourself with good minds and learn, and just direct the ideas. Don't interfere, but manage and be a good leader in this regard. Yeah.
No, absolutely agree. I think it was also Steve Jobs definitely said something similarly. I think so. Okay. But also I think I remember also Jack Welch from GE. It was already in the 80s, 90s, where he said also something similar, that everybody on your team or on the leader's team must be better than the leader in one or another aspect. So if the leader is the best solution for everything, the leader is doing something wrong.
Exactly. I fully agree. I couldn't agree more.
Now let's talk about success stories. What were your 2 biggest successes that you achieved for your customers?
Okay, for the biggest success for our customers. Okay, so I have to think about what I am able to explain and what I'm able to display.
So non-confidential information, or display, you mean, do you want to have a presentation that you want to show?
No, I really think about confidentiality now. So I think we know about several molecules that we have co-developed, or let's say, no, co-developed would be too much, but we helped to develop them at some stage, which are now in clinics. But I think one of the success stories that I have in mind was with a company, a biotech company, again, early stage VC backup company on the protein, ProTech realm. And yeah, basically they didn't have large capacities and knowledge in biophysics, but they had really a push from the VC to speed up and to give results and solutions. And the time frame was very limited, so it was not much time to do. And it really started with a hit identification so screening of several hundred molecules to identify a potential hit to the protein of interest. And we managed to do that in an incredible fast time. We were really skeptical if we could make it until the deadline that we were passed to. And so they were absolutely fascinated about this. The VC was absolutely astonished about how we could manage to do that. And in the end, it turned out that the process or the project was even continued much longer, for nearly half a year longer, with MedChem optimization rounds, with all necessary tools. And we always were helping and could help here. In the end, this molecule is now in phase 2, and it's absolutely
cool. Congratulations!
Congratulations! Really cool. Of course, our contribution was probably smaller, but these guys made an incredible job anyway. So they fully deserve their success, and it's cool that we have been a part of that. It was really nice.
Yeah, maybe we can talk about which company it was later in another episode. So,
okay, another success story. I mean, one thing that even people could look up on, maybe that's an offer that I can explain, is a formulation study that we have done with an HIV vaccine candidate. So it's a rather internal project, so I'm happy to share a lot of information here. It's not confidential anyway. So there's an application note that you could even download on our homepage anyway. So it's a collaboration with university clinics in Regensburg, the group of Professor Wagner. He's an expert in HIV, studying that virus since many, many years, and doing a lot of effort in vaccination, because obviously still there's not a good vaccine in the market that helps for that disease.
I'm interested in your opinion. Sorry to interrupt you. I mean, mRNA, for example, for solving SARS-CoV-2 was a pretty quick shot. So I mean, I know the development was much longer from the technology. It started in the 60s, and it's not in one weekend. But basically, I think Moderna and BioNTech had in a few months a solution to a problem. Why is it so challenging to develop a vaccine against HIV?
I think there are efforts. And I don't know exactly what the status in the moment is about mRNA vaccines about HIV, but I know about efforts that are done there, and I would not be wondering, or I would not be surprised that at some point there might be a success. The missing piece of vaccination is simply because in the past there was the basis was always an envelope protein of the HIV, and here it's very challenging. So the approach was simply maybe not the correct one, or correct I wouldn't say, but it's challenging. And now, honestly, with this revolution that we have seen with the mRNA vaccines, I see a lot of doors opening up, and you're fully right. I mean, maybe mRNA is a solution also for that. But I would definitely recommend to talk to an expert in HIV.
I was just curious. Exactly. It was not emphasizing mRNA, the potential in HIV. I'm just curious because I think HIV now, vaccine development is about 20 years, 25 years, something like that. And we have no vaccine yet.
Yes, correct.
And the mRNA example I just brought up because we have a solution for SARS-CoV-2. I mean, some people say it's not working well enough. Some people say it's working better than expected. Some say it's okay. But we have a solution. And for HIV, I think we don't have a solution yet. Why, just interested in your opinion, why it's so complex to find anything against
I mean, the reason why the former vaccines had troubles is, of course, because it was really based on the protein itself. So the envelope protein, it's a trimeric structure which is then composing the HIV virus, is of course decorated with large glycan chains, so large sugars, which make them very hard to tackle by the immune system. So this is why this guy is always escaping the immune system. In the same moment, it's difficult because if it can escape the immune system, how can you then induce a certain vaccine reaction, right? So this is the challenge here at that point. So you would have to produce a certain protein that is so similar to the original HIV envelope, complete structure. And here this is a challenge. So there are efforts based on broad neutralizing antibodies, which are very fascinating molecules because they are antibodies, but they have absolutely fascinating structures, very long, spiky things that can really dig into this forest of glycan chains and reach the center of the protein or reach the protein surface. Absolutely fascinating molecules. But I think there's a general topic about immunogenicity of this virus. Again, without being an expert in this. But I think you're right. It could be that we're talking about a problem which is maybe not even a problem anymore, because any good maybe a group in the world has already worked on the mRNA for HIV. I'm not up to date on that.
Yeah, but I'm just coming from the financial point of view. So I have an economic background, and I always think, I mean, if we had the same dedication to cure all diseases in the world that we had for SARS-CoV-2 in the last 3 years, I think many would have solved many problems already. Maybe this is also a call to action for politicians to also look at other diseases, because there are still unsolved problems that cause unnecessary suffering, in my opinion. We have great science, we have great scientists, just need funds.
Yeah. I mean, honestly, I think that's it. I mean, for example, if we are already on that interesting topic, I mean, also antibiotics is a big topic that is difficult to solve by the economy. So there are now the first, approaches where even countries make together some efforts to create new antibiotics. And the reason is that an antibiotic cannot be sold too often, and then it's financially not attractive to be developed. And this is simply a pain. This is the origin, is the basis of all what we do. It's the capitalism, basically. And here, I think there are solutions. And I think, as you're right, there are some diseases where I think a more official approach, not economic approach, but let's say by funds or by countries should come into game and help to fight against these things. Because for some of those things, again, the approach via the open market might not be the right one. Yeah, difficult topic, but I'm not sure if I'm right here again.
Let's go back before we come to drug development to the formulation development. I interrupted you with my question about HIV. No, what is the role of biophysics in developing formulations for HIV vaccines?
Yeah. So what we have achieved here was basically to stabilize the protein, which is called BG505 SOSIP. Is a stabilized HIV envelope protein designed by different innovative groups in the world, academic origin. And Ralf Wagner was also part of that development as one of the consortium partners, together with Nanotemper Technologies and us. And we basically found conditions where the protein is highly stable, where the protein is not falling out of solution, where the protein is not aggregating, where it's stable over a certain period of time, and really can now be used in that buffer system. So you can imagine it's a protein that is swimming in a soup, and the soup has to be with a certain combination of ingredients. If it's too much acid in there, for example, then the protein will not be happy and will not be will already be destroyed once you reach the arm of a patient or whatever, how you inject it. And you have to have a certain salt, for example, a grain of salt here and things here, and you have to combine that to find the right soup. And the soup is then supporting the protein which is swimming in there, let's say. And the soup is called formulation, basically. So the formulation is a very important aspect about how to use such a vaccine candidate. And we did a lot of tests, for example, for stress testing. Does it survive certain stress situations? You can imagine the vaccine candidate is simply not kept in a cold chain. So somebody forgot to give it back to 4 degrees. It's there on the bench table for a day. What happens? Can I still apply it? Is it still valid, or should I discard it? What should I do? And we can tell, okay, by checking different physical parameters, the protein has survived this. It's no problem. You can still use it. And these kind of things we have done, and we came to a really nice formulation. And I'm also not absolutely updated on the current status of the project, but I know that the plan was to bring it into the first animal study.
Cool.
Based on that formulation. So it's really cool. We were very, very fascinated to be helpful in that regard of bringing a HIV candidate really into the first animal study. Really cool story, I think.
So you can take an existing formulation and make it better. Basically,
yes.
understand it more,
correct,
and help improving the end result.
Correct. Absolutely.
Whatever the goals of your customers are. So it's flexible. It's flexible. It's not a one goal company. So basically you ask for the goals from your customers and then help them to achieve their goals with physics.
Correct. That's correct. And that's
I was just smiling before because I said the soup. It's a formulation. It's called soup. So is it a technical term? Soup?
No.
Can I use it next time?
No, please don't use it. It just came into my mind to make it, let's say, more digestible.
Yeah. No, I like it. But next time when I talk with a scientist, they will say, "Well, which soup are you cooking here?" And I will call you out. So.
Oh God.
I learned it from Dr. Schubert.
Oh God. No, it's really just a fun thing. So it was just digested better.
But soup is really interesting. Formulation is, I will use it. It's funny. And we should make more fun in the world better, I guess. So we should have more fun, generally speaking.
Absolutely.
The project HIV development, and you said you started your company in 2011.
Yeah.
I'm now in the life science industry, pharma industry, since 2006. My first attempt to help scientists moving their science forward was also in antibiotics. It was a Novartis spin-out.
Okay, cool.
It was a company called Nabriva, the department. Rotger Novak built the department, who later founded CRISPR Therapeutics with Emmanuel Charpentier in Switzerland. A lot happened in Vienna, but ultimately, and this is the point of the next row of questions, the scientists leave Europe for the United States because there is just more funding on the market than we have in Europe, which is a big downside. I had recently on my podcast Sebastian Malaby, do I have his link here? Big shout out if you want to understand venture capital, the power law. He researched all the backstories to the development of venture capital, and a huge part of this book is Genentech case. Which is pretty interesting. Bottom line is we don't have much venture capital here in Europe. And when I started in the life science industry, I had really the advantage to work with a great company, with a great spin-out, with great scientists. But there was almost no zero landscape on the market. So in 2006, we had to build a lot. So we also had animal facilities for a small scale. I mean, it was 60 scientists in a company with own animal facilities, which nowadays is out of scope for any VC. So they just say, okay, use a zero. I know how I experienced the development of the life science industry in 2006. I'm curious to hear your perception from 2012 or even earlier. How do you see the development of the life science industry in Europe in the last years?
Yeah. So first of all, I agree. A lot of good ideas are unfortunately leaving Europe. Unfortunately, also dramatically leaving Germany and Austria, as you mentioned in Vienna. It's a great hub, Munich as well. And a lot of companies or a lot of ideas have left. This is a real pity because the idea is not the product in the end, and what you in the end get back is not in the place where the idea was thought of, and this should be changed. And one of the big reasons, as you described, is the money landscape, the VC landscape in the US. Since I was in I'm also part of BioDeutschland, which is a union of biotechnology companies in Germany. And since I'm there, I know that these guys made a lot of effort and talked to politicians to let them understand what the problem is and how much taxes they leave simply because they do not make an attractive situation for an investor in Europe. And this has taken a lot of effort of what they do. And I know there's slowly things are changing. And at least we see that in Europe, a lot of, or not a lot of, but they are now getting more and more interesting companies, also interesting funding, maybe still not as that level as in the US, but at least more as expected. Of course, in the moment, I think we have a special situation with the COVID situation where a lot of money is pumped into general every market, with the consequence that a lot of, or some money was probably not placed at the right spot with the right idea, which we now face as a natural correction of that phenomena. Then we have a terrible war going on in the Ukraine, also affecting the complete economy and also affecting, of course, VC funding landscape. But at least from our perspective, is that we see that VCs may have more, or may take it more serious to select the right topic and the right project. If you look up the latest Series A, Series B, Series C, which circle around, there's still money in the market. But you see first signs that people take it a little bit more serious in which type of effort they invest. For us as an outsourcing partner, the landscape is a little bit different, because obviously in this difficult situations, let's say, a variable cost factor, outsourcing, is sometimes in favor for an internal fixed cost factor. Mm-hmm. And people then would either postpone internal investment or even, don't take the investment at all and favor outsourcing. So this is our situation. It's a little bit different to the classical in the moment VC situation. For us, the situation, in my eyes, it would kind of balance out. So in the end, my personal opinion, right? Again, my personal opinion is that if even the market has overall a little bit less money, the outsourcing is still increasing and that levels out for us. So this is how I expect it. And I think I have done a lot of conversation with other CROs, from medicinal chemists to mass spectroscopy guys to NMR guys and so on and on in Europe and US. And basically the information and the opinions are more or less the same with smaller modifications. So in general, there's a trend for outsourcing, which is clearly seen since the last 10 years. Reasons one is specifically now for biophysics and also for sure for other specialty niche areas is the lack of skilled people. Mm. So for a biotech company, it might simply be difficult to find the right people and to even keep them for a longer period of time. You don't need a person which is off after 2 years. You need somebody that is constantly for a long-term period with you and supports the company. This is one big challenge why outsourcing to expert groups, I think, is in favor in the moment. And of course, also the investment into the machines, into the complete processes. Sometimes it's clear that the investment into other areas is simply more interesting. And therefore, outsourcing is in favor in the moment, definitely. You can see the market reports talking about several increase in market expansion every year. I'm not sure. I think it was 5 or 6% per year. So the complete market is growing dramatically. So for outsourcing, and that is also what we see. And I think it makes a lot of sense. People specifically biotech companies, they were built up on a certain knowledge and they are really good in their knowledge, and they should take certain processes and bring them to experts and let the experts run that thing. I think understandable.
I couldn't agree more. I was just, while you were talking, I was just looking for a book because it was my research focus in the 90s. But I don't, it's from St. Galla here. It's Networked Economy. Das Netzwerkunternehmen. It's
Okay.
Elgar Fleisch wrote it. He's at the University of St. Gallen, I think, meanwhile, ETH Zürich.
Mm-hmm.
And I think the 80s, 70s, 80s, 60s were different because companies hired people for everything.
Mm-hmm.
And with the knowledge society, as you said, I mean, when you need an expert once a year, I think companies who hire an expert for one consultation per year, even when they can afford it, break the expert, because what is the expert doing? He's sitting around. So it's just 1 year, 360 days sitting around, and 1 day of work. It's like destroying a racehorse. So if a racehorse is not running races, it's just getting weak. And outsourcing, especially in a specialist economy, absolutely makes sense, because when you focus on biophysics, on biophysics, you can throw challenges on your team every single day. And this trains the people, and it helps them to get better in their field, compared to a company where they get 1 day of training per year. So over time, your team will be more successful because they just gather more information, they get more experience, they can improve. So outsourcing makes a whole lot of sense for everything that is not needed on a daily basis in a company. And luckily, with the modern technology we have right now with Zoom, for example, we can now do Zoom calls. It was unthinkable 6 years ago, or even 3 years ago. You can also engage with experts somewhere in the world. When you have a group, can catch them on a call, meet them once, and work together then virtually.
Absolutely. And that is exactly, we have showcases. We work with pharmaceutical companies that have awesome equipment, latest stuff that we also hear is great, but the key personnel has left. Bam. What do? The great and really cool process that they have bought up, built it up, suddenly is down. The key personnel is gone. So sometimes our task is just to coordinate how to build that up again. That's exactly why outsourcing is important. So to avoid these situations, this is a pity for them. They have invested so much money and effort and time into building up the complete process, and suddenly one piece is missing. And that is why outsourcing makes sense a lot. But in the same moment, one also has to be very fair and to say, listen, if you use a certain process every day, please think about integrating this internally. We will help you with that, because that is something that you will use every day, and you should have that under your control. That's the opposite thing. And I think we should see the both things of both ends, and at least we support on both ends.
Yeah, I totally agree. It's similar like the social media trend, for example. I mean, it's something that many companies also in life science are now doing it. 3 years ago, nobody did it. Nobody wanted it. And also my suggestion is, when a CEO is not sure, let's just stay on the social media example, is not sure whether they should do something on social media or not, get an expert, pay an expert. So If you pay them an hourly rate for consultation and you find out after 10 hours it's not your thing, nothing is lost. So you just cancel the contract and say, okay, thank you very much for your help, but we now do something different. And if a CEO says, okay, I would like to test it further, then he still can stay on an outsourcing level and say, okay, post something for me or come up with suggestions once a week. And when the CEO then sees that things start working, that it really makes a difference in the company, and he needs the consultant every single day, it's the right point in time to think about integrating the process into the company and make sort of social media. I know tech transfer is not used in social media, but it's basically transferring the know-how, the expertise into a company to an employee. And also when a company is growing, I think for big pharma, it would be stupid sometimes to outsource parts when they can just build their own department, especially social media activities. Just imagine Pfizer, they don't need outsourcing partners. Probably they can build a huge department internally because they need it on a daily basis anyways.
Sure.
But for the small biotechs, when they need, let's say, a press release every 6 months, and they want to do it with audio and video, probably it's better to find an outsourcing partner who stays up to date with technology, who knows how the platforms work. And I think it's similar in your field. I mean, when someone is not sure whether biophysics can really help speed up the development process in the company, it's good to start with a consultation and try things out, play around a little bit, be playful. And if it works, integrate it. And if it doesn't work, just stop it.
Exactly. Absolutely. And I think that's you can transfer that to different things. And I think your example of the social media is absolutely brilliant, because that's again one of these things that you probably have not learned as a CEO to handle these things, right? You don't have the education, but it's also something where an expert should help, because it's you can do a lot of things good, but you can do it better if you're an expert. And so I fully agree with you on that.
Yeah, with experts, what I think is good with experts is the focus to have up-to-date information and not outdated information. When an expert can't train on a daily basis in a company, I think this 10% of something, so having someone in a company who is 90% engaged with other things and 10% with something might sound like a good idea initially, to say, okay, we have this 10% role in the company, but this person doesn't get any training. While a biophysicist in your company, what should they do? They just need to work every day on the same topic. They need 100%, they get 100% challenges.
Absolutely.
And this makes a difference on the long run.
Absolutely correct.
Let's talk a little bit about trends in drug discovery and drug development that you see, especially with a focus on biophysics. When we look into the future for 10 years, it's the fun part. I know predictions are very, very hard and very tricky, and they might not come true. But let's just play a little bit around with it. With your expertise in biophysics, where do you see drug development and drug discovery in 10 years? And where do you see the role of biophysics in that field?
Okay. So first of all, I think to start with the second question, biophysics will still be very important because at some point people will need a validation in the lab for anything, whatever they have found. Now, coming to the trends, I think there are several trends that are already visible. Some of them, I'm not sure if you even can call it a trend anymore. It's already kind of even settled, kind of. But for example, the in silico prediction. So it's really something that started 5, 6, 7, 10 years ago, I would say, to basically give the computation a chance to predict a certain drug. And there are different approaches now also strengthened with AlphaFold and also some other interesting algorithms to predict structures of proteins. This is definitely still an emerging field. I think one has to be fair that so far, with some nice exceptions, I have to admit, this concept should still show the efficiency a little bit more prominent in my eyes. That's my personal opinion, right? With some exceptions, though, there are great companies out there that do an incredible good job. but it seems that it's not that easy as everybody said. So you cannot simply take a random algorithm and simply modify it a little bit and then predict the best compound ever. You need to be an expert in that. So this is, I think, the consequence of what we've learned in the last years. And this is something that has not been there in 10 years before. It's now something that is modern. And I think that is something that is even more pronounced in future. So I still am convinced that this is something that will give more or will gain more attention even in 5 to 10 years. So giving the chance to a computational prediction to identify compounds or to help to develop drugs. Reason one is that the algorithm is getting improved and improved. Reason two is that the speed of computational capacities are increasing a lot. Quantum computers are for sure in 10 years a topic that we can discuss. And then, of course, with higher power behind computation, you could even reach more things. So I'm still convinced that this has a fair chance. Still, in the end, you need a certain validation, and biophysics is one of those validation tools where you can then see if the predicted compound will bind to the target of interest, whatever it is. In general, I see that my opinion is that in 10 years, the landscape of the classical small molecule-based drug discovery has changed a lot. I think small molecules will not be the entity of future. I think PROTACs will take over a large part of the market, and also biologics. Biologics are on the run in the moment. I'm not just talking about antibodies, but other entities which will take more and more market share of the drug discovery realm. This is my personal opinion. And I don't think that the classical small molecule drug discovery has that large share in 10 years as it has now. Yeah, so I think that's the summary of what I think are the trends. I also think, I missed one thing. I think that the focus will also be more and more on structural biology and giving structure more relevance in drug discovery. Meaning medicinal chemists will take this even more into account in future. And then, of course, also related to the general topic of other life science areas which take more or will be more important are the systematic approaches to really see the cell as a system with interconnectivity. And I'm very sure that in 10 years, the interconnectivity between factors will be more and more relevant as it can be today for different reasons. Also, for example, computation and these things will improve that. So I think, therefore, in 10 years, drug discovery will be different. Different entities, different approaches, different molecules that are of great interest. Bottom line is, however, that biophysics will still be playing a big role.
Yeah, I believe that you mentioned at the beginning simulation and the switch from chemics to biology.
Yeah.
I didn't find a graphic. I was hoping that I find something quickly while you were speaking. I think this trend is there. First time that I realized it was 2015, 16. Biologics was, I think, a very small part of the market 10 years ago, and now it's just growing. And I don't know if it's correct, but I think it's 50-50 meanwhile. It's I'm not quite sure, but
I also I would agree with that number, even that I don't know it, but I think that could be the case, yeah. So at least what you see when you compare the FDA approvals, when you look at the small molecules and biologics, they take off and are really boosting much more than small molecule-based FDA approvals.
And the third part you said systematic. I was thinking about systemic approach. So one of the questions I had early in life was how do disease evolve? So how do diseases start, and what can people do to stay healthy in generally speaking? I'm also hoping that in 10 years we integrate more early stage changes in the body. And what I find really great these days is that a lot of scientists spend time to investigate how lifestyle, for example, also changes the biological system that, let's say, encourages the body to stay healthy. And science can make it just better together with the pharma industry. I mean, there are so many things playing around with nature. What I heard very often in the last couple of days, and also with a few recordings, was which I have hard time believe, to be honest, simulating the entire drug development value chain. So it's just putting a compound, biologics or small molecule, through a simulation and do all tests and trials within the simulation. What's your opinion? It's just guesswork. Yeah? It's not science anymore. It's just the fun part of the conversation, where we can just bluntly guess and label it as it's a best estimate and just making fun here. Do you think this is possible, that at one point in time with quantum computers, it will be possible to simulate everything to speed up the drug development process? So it's just putting an idea into a computer and let him or her do the calculations, and at the end of the day you get your new drug out of a out of a factory. Now I have it.
So let's assume this is true. I would say that human nature will simply not allow that a computer will just do everything, and we want to see the proof. So I think that at some point we will need to see the proof, and at some point it has to be validated. And at least in the later stages, I doubt that any prediction can really come up with a tox study. People will want to see this. They want to see how a cell is acting when it really touches that molecule, whatever it is. I doubt that in this point latest that any human nature would allow a prediction to be as valid as an experiment, because here it's about life. And I think also the authorities will simply not accept this. I would agree that in the first phase, drug discovery, very early one, I could imagine that a certain level of automatization is there with predictions, because nobody's harmed if it's wrong. But latest at the point where a human being is harmed or could be harmed, or a cell could be harmed, at that point there has to be a proof. And at this point, the prediction is out of the game. That's my personal opinion.
Yeah, I come from the economic side, and I calculated a lot of predictions in really simple models. So the capitalistic system, so we were talking about it earlier in the conversation, predicting the future of economy of a company in a stable economy is pretty easy. You have a few parameters you can fiddle around, when you just can assume that the customer demand will stay the same. But it's impossible to predict disruptions. For example, was in the mobile industry for a couple of months in 2004-05 to make a company more attractive for a takeover. And nobody, I think, in the industry here in Europe expected that Steve Jobs will put a smartphone on the market that just destroys all mobile phone producers here in Europe. This is unpredictable. When I think about biological systems, they are very complex, and a lot of things like sleep, like nutrition, bad relationships, just change the composition of the body slightly and make the body more prone to become sick. To simulate that, these subtle parameters that have an influence on the biological system in a computer, I have a hard time imagining it.
Me too, honestly. Even though I do come from the epigenetics realm, so the regulation of genes on not on a DNA basis, but on a level above the DNA, and so it's proven that there are several regulations which most of the time come over generations, not necessarily in an organism itself, but maybe in the siblings, where a certain change has happened. And it could be bad nutrition, it could be these kind of things. So here there are absolutely proofs, and I absolutely believe in that this is important for you and also your siblings to be healthy and to take care about that. I also do believe that it also has consequences for yourself. I believe that. but I am sharing your opinion. I am not sure if you can simulate that kind of complex system as a cell is. I know that climate is also extremely difficult to predict. I know that a lot of things are highly difficult to predict, but I still believe that a cell, I mean, you have several billion cells in your body and they each interact with each other. And so there's so many overlaps, and I simply, maybe I'm too short in mind to really believe in that, but I don't.
Yeah, maybe with quantum physics, the funny thing was that it was in the second half of the 90s when I was talking with martial arts teachers about how to stay healthy.
Mm-hmm.
And their recommendation basically was drink more water, sleep more, and exercise. And have positive social relationships, and of course hygiene. So it was basically these 5 elements. And it was not enough for me. So I said, where is the proof? So where are the studies that prove that what you say is right?
Yeah.
And there was none. I mean, the late 90s or the mid 90s, there was this time when the internet was not really very evolved. Getting studies from somewhere in the world meant asking a lot of questions to people to find the right experts who is doing a study. And now we have Google. And whenever I want to find a study, I just ask Google and say, give me a sleep study. And the great thing is that what they said back in the 90s through observation, basically, through centuries of observation in martial arts, is now proven right by scientists that in studies, it's a little bit later, to simulate that. I don't know if it's possible. So when you go down the disease route, then how does new small molecules or any biological approach affect a system for the first time? There is no information there. So with sleep in martial arts, basically they had centuries of observation, centuries of data, simulating something new when you throw a new small molecule into a body.
I think there's also maybe this topic about teaching an algorithm with existing data and creating data. I think there's a discrepancy, right? So I'm not sure if you can create something new. You can teach on basis of observations, as you explained, but I'm not sure how the translation would be to create something new then. If we call about this drug discovery, prediction things. I'm not an expert in this, but
I think it was Jack Ma from Alibaba who said that we should train our children in creativity because creativity cannot be simulated. Everything else can be automated, but not creativity. And it would mean, in my opinion, it's a philosophical question maybe, can AIs be creative? Can they be trained to be as creative as humans?
I would love to have a conversation with an AI guy about this. I don't know, but I think I'm not sure. I'm not sure because it's an algorithm that learns from something. But there's a fundamental difference between learning and then having an own effort to be creative. There is a difference. Not sure, but that would be super interesting. You should get somebody from the AI world and discuss that. And I will listen to the podcast several times. I promise.
If you know an AI guy, I mean, I know one is
yes, actually, I also know one.
Send him or her my way, so we can have a conversation.
I'll do that. That would be. I mean, it's a really fascinating philosophical question. Absolutely.
I mean, I loved playing when I was in school. It's the 80s. I loved playing chess. And this was also the time when the first chess computers arrived on the market. And then I stopped for 3 decades. And for some reason, I think I saw a podcast about chess. I'm now interested in chess, and a lot changed these days. And there are a lot of bots on the market. There are a lot of chess computers, chess programs on the market who are even better than human beings. But it doesn't impress me. So when I see, okay, this is a bot or a computer who can beat Magnus Carlsen, for example, the best chess player currently in the world, as far as I observed it, it doesn't change anything. But what created motions in me was this discussion between Niemann and Magnus Carlsen, whether this other chess player, Neumann, this other chess player cheated in a game or not. So yeah. I find this very attractive when two people want to find out who has more skill in a certain area. And I admire people for having the focus and the time and the nerves to become proficient in one area, in one walk of life. This I think is attractive. When a machine achieves something in that, for me, it's not very attractive.
Yeah, I think it was from the first instant, I have to admit that I was very impressed. So I think Deep Blue was the first of those chess computers that was beating a chess master,
Karpov also.
Or Kasparov, or how, yeah. I think it was a Russian chess player. So I was impressed because it was like in the, I don't know, 90s, 2000 maybe. I was like, wow. The machine, the robot is better than us. So it was the first time that I thought, okay, something that we created is better in something than we are. But on the other hand side, a car is also faster than you, right? It drives faster than you. Or there's so many things that are better than we are as human. And then I thought, yeah, okay. So here it was so fascinating because it was the first time that something like from the mindset here was better than a chess player, a skilled one. But I still have some kind of fascination about this. But I do understand your point. It's more interesting if humans, which are on the same level, discuss about that, because at some point you can train very likely every robot to be better in some aspect a human has.
Except creativity. I mean, Arnold Schwarzenegger, Terminator, was also in the 80s, so maybe we will see. Thomas, I could continue having fun with you and talking about various aspects of life. I think we are drifting off in the fun part of the conversation. Let me ask you the two final questions. Is there anything that you would like to talk about in this podcast that I didn't ask you so far?
No, I think it was great fun. You asked really interesting questions, and I have nothing that I really wanted to add more. I think we found a good solution of having discussed certain trends, certain landscapes, general thing. Also giving Tubind and the company a certain room. I think it was very nice. So I enjoyed it a lot.
Me too. I had fun in this conversation. Then my last question is, when someone who listens to the podcast or watches the video on YouTube or on LinkedIn or on Facebook later on wants to reach out to you to engage with your company to find out whether your solutions are the right solutions for their companies, what's the best way to engage with you?
So we are a young company, so social media, homepage, these kind of contacts are easy. There's contacts form that you can easily reach out to us. The info at tubind.com email address, it's easy to reach out. But of course, we are also open for classical calls, phone calls, and I'm really happy if somebody reaches out to us. I hope I could give the impression that we are open-minded staff here. And I promise that we are, first of all, listen well, and then we will try to help as good as we can. That's what I promise. And yeah, please reach out to us. It's a pleasure.
Fantastic points at the end. So you're a modern company, also starting the explanation how to reach out to you via social media. Are you still doing real life meetings as well?
Yes. So I think that's still very important. I think having the direct there has to be a certain chemistry between people, right? And for both ends. So, I mean, we also enjoy working with people. And for us, it's also important to have the right chemistry with the people, not just the people with us, but we also. So there's a give and take. It's a relationship between people. And so both both sides have to be a certain comfort in talking to each other, being open, being transparent, being honest with each other. And this has to be built up. And of course, meetings in person have still, I would say, some level above the social media. So having a handshake, having a good conversation, and these things, it's important. Absolutely.
I couldn't agree more. We need both things, social media and real life. A good mixture of that saves time and creates connections and relationships. Thomas, thank you very much for your time. Thanks a lot for this great explanation about what biophysics is, how it evolved and developed in the last 10 years, and future predictions. I wish you and your team all the best for the final days of this year. And I think can we say already we are for Christmas. I wish you a Merry Christmas and a great start into 2023. So it's the first time that I wish that.
Okay. Thank you, Christian, a lot for the really, really nice open discussion. And it was a great pleasure and a great concept that you have. I really enjoyed it. Thanks a lot. Also,
thank you very much.
You have a good time.
Thank you. Have a great day. Bye-bye.
Ciao.